Hello everyone, my name is Dr. Avelyno D Costa, and in this video I will be talking about DNA repair. This topic is actually divided into two videos because there are a number of examples that we have to cover. In the previous videos we saw about the different types of DNA damage. So under this topic we will be seeing how some of these damages can be repaired by certain DNA repair mechanisms. So, cells, when they have observed that their DNA has been damaged, they try to repair this damage as fast as they can, such that this damage never ever gets propagated into the daughter cells. So, they will recruit a number of DNA repair enzymes and machinery to repair this damage. Such that the damaged DNA is repaired, and then only will the cell be able to undergo mitosis or any other type of cell division processes. However, if this damage is too high, that is, if it is too much for the cell repair machinery to repair, then the cell usually programs itself for death, that is, it will undergo apoptosis. So, this is a better option compared to taking the effort to try to repair the DNA because that much energy and that much time would be required for repairing the DNA. It would be better for the cell to kill itself. So it's kind of like a fail-safe mechanism by which the cell ensures that damaged DNA never ever gets propagated to the daughter cells. Because if this happens, if damaged DNA gets propagated to the daughter cells and in subsequent generations, it can actually lead to mutagenesis and it can lead to carcinogenesis. So the cell ensures that such damaged DNA never ever gets propagated forward. It tries to repair it as much as it can, but if it sees that it cannot do it, then it would undergo apoptosis. So this repair happens in certain checkpoints of the cell cycle. As you are aware, there are a number of phases of the cell cycle, and between each phase is going to be a checkpoint with which the cell checks and ensures that everything is processing smoothly, that everything has been doubled correctly, if all the DNA has been replicated properly. If there are any errors during this phase, then this cell cycle is going to be halted. The DNA is going to be repaired, and then only will the cell undergo the cell cycle and subsequently mitosis. So there are a number of genes and number of proteins which help to arrest the cell cycle such that the DNA repair machinery can come and repair the damage. So we have a number of these proteins which are coming from the genes that are the tumor suppressor genes, and we must be aware about some very important tumor suppressor genes that are there in our body, such as P53, P21, PRB. These play a very, very important role in halting the cell cycle such that the DNA can be repaired if damaged DNA has been detected by the cell, and only when this damaged DNA has been repaired, then only will these tumor suppressor proteins kind of will allow the cell cycle to progress forward. So under DNA repair, we have different mechanisms under which you can see some examples. So we have for single-stranded repair, we have three mechanisms that we can talk about. That is the direct repair or the light-dependent repair. Then we have the excision repair and the mismatch repair. And under the double-stranded repair, we have two types, that is recombinational repair and non-homologous end joining. In the first one, that is the direct repair, there's a specific type of direct repair which you can talk about over here, which is also referred to as the light dependent repair. So in light dependent repair, this mostly occurs in bacteria and in certain eukaryotic cells where pyrimidine dimers are specifically going to be repaired. So pyrimidine dimers, as we have seen in the previous videos, that these are two pyrimidines or thymines that are there on a single strand close to each other, and because of the presence of UV light incident upon the DNA, it causes the formation of some covalent bonds that causes these thymines to come closer together. That will lead to a distortion of the backbone. So this will lead to a distortion of the DNA helix also. So there is an enzyme which can help to repair this type of damage, which is referred to as DNA photolyase. Now, DNA photolyase requires light in order to repair this damage. So that is why it is referred to as light dependent repair. So photolyase takes the energy from light and helps to break these covalent bonds that exist between the two pyrimidines that occur in the dimer. So it will break this covalent bonds, and it will return these thymines back to their original configuration without these covalent bonds, and then the structure of the DNA will go back to normal. Okay, so you can see that if these covalent bonds, they need to be broken in order for the DNA backbone to return back to its normal configuration. Another type of direct repair is that involving the enzyme called O6-methylguanine DNA methyl transferase. And this particular enzyme helps to repair alkylated bases. So if you remember in one of my videos when I spoke about the DNA damage, is that there are some chemicals which add an alkyl group onto the bases. And one particular base which is created because of alkylation, when an alkyl group is added to guanine, it creates a base which is referred to as O6-methylguanine, which mispairs with adenine. When guanine is actually supposed to be pairing with cytosine, O6-methylguanine mispairs with adenine. So an enzyme is there called O6-methylguanine DNA methyl transferase, which directly removes this alkyl group and restores the base back into guanine. So that is why this is referred to as a direct repair. Then the next type of repair is the excision repair, under which we have two types, that is the base excision repair and the nucleotide excision repair. So first we will see the base excision repair. So under base excision repair, there will be a damaged base which has been incorporated probably during the process of replication. So it is going to be a modified base or a damaged base. And then what is going to happen is that there will be a set of enzymes which will help to remove the damaged base along with the nucleotide. So what happens first over here is that the base is going to be recognized and is going to be removed. So who does it is an enzyme which is referred to as DNA glycosylase. So DNA glycosylase will recognize the damaged base by some mechanism, and it will help to remove the damaged base. Once it removes the damaged base, it creates an apurinic or an apyrimidinic site. So you can see only the base has been removed, not the entire nucleotide. Okay, so first the base is going to be removed, and once the base is removed, it creates an apurinic or apyrimidinic site depending on what type of base has been removed, whether it's a purine or a pyrimidine. Then in the next step, another enzyme comes and cleaves the nucleotide after that. So you can see it's a stepwise process. First the damaged base is removed, and then the deoxyribose sugar is going to be removed after that. So an enzyme comes which is referred to as AP endonuclease, which cleaves the phosphodiester bond on the 5' site of the apurinic site or apyrimidinic site, and it removes out this nucleotide. Okay, it removes out this sugar. So you can see a big gap that has been created over here. So these two enzymes help to remove the damaged nucleotide. Okay, in a stepwise process. First the base is going to be removed, then the nucleotide will be removed, and after that DNA polymerase will come and it will synthesize the correct nucleotide depending on the template DNA, obviously. So as you know, DNA polymerase plays a very important role in synthesizing DNA, and there are a number of polymerases which are involved in the process of actual synthesis, proofreading, correcting damaged DNA, right? So one form of DNA polymerase will come and it will read the template strand and will determine what base has to be, what nucleotide has to be incorporated over here. Obviously the NTPs, the specific type of NTPs, whether it is ATP or GTP, CTP or TTP, it will go and it will bind over here. And if you remember the process of the phosphodiester bond formation, the phosphodiester bond will form over here. A gap will be sort of created at the 3' end. So DNA ligase will come and it will seal that nick, or it will seal that gap. Okay, so DNA polymerase plus DNA ligase helps to replace and seal the nucleotide back into the DNA. Okay, so you can see in this way how the damaged DNA or the damaged nucleotide has been excised, it has been removed, and the correct nucleotide has been synthesized and correctly incorporated into the DNA. Okay, so this is one type of excision repair which is referred to as base excision repair, which works to remove out the nucleotide in a stepwise manner, and the correct nucleotide is going to be restored over there. In the next video, I'll be explaining to you about the other type of the excision repair, that is a nucleotide excision repair, and some other types of DNA repair processes. Thank you.